something has happened. But you may not realize it because it's very subtle. It's hidden in all the data. That's the way we run medicine today. You wait for a disaster to happen, and then you get treated. Like I said, we have 5 variables or so we consider to be vital signs, which are just very basics, but now we can maybe get thousands of them. So how is a physician or biomedical researcher supposed to keep that in their head at the same time? But with AI, it has this remarkable ability to, at the same time, think about thousands of things simultaneously and put them all in context of one another. We can't do that.
Hi, Professor Palsson. For our listeners who don't know you, could you please briefly introduce yourself and talk about your work?
So let's start from the beginning. I'm born and raised in Reykjavik, Iceland. Got fascinated by chemistry and biochemistry early in my life. So I went to the university to study that. And during the summers, I worked on isolating enzymes from rat hearts and studied their kinetics. And this was a fascinating subject, but I realized that one enzyme is not informative about the living process. There are hundreds, if not thousands of them working at the same time. And I came to this realization that cells are systems. We study components, but we don't describe the cells as systems. Those were my initial thoughts about systems biology, or what is known as systems biology today. So I tried to find a major that could help educate me in the chemistry, biochemistry, as well as the mathematics, because you need mathematics to study complex systems, right? And therefore I went into chemical engineering. I went to the US, got a degree in chemical engineering, got into biochemical engineering, then more and more medicine, so biomedical engineering, and now I've spent 40 years as a faculty member in the US, 10 at the University of Michigan and 30 at the University of California in San Diego, with appointments in engineering and medicine along the way. So that's kind of how my career unfolded in just a few sentences.
You figured out how to digitally model an entire living cell. What does that actually mean, and why should a normal person care about that?
And how much time do we have? This is a very complicated task. Today it's called digital twins. You have a real cell and a digital cell, and over time they will become identical. So you can actually simulate in a computer what the real cell is doing. And so when we started doing this process, was a very humble beginning. We just could capture a little bit of what the cell does. And we are now reviewing this 40-year history, and it has unfolded in 5 distinct phases. The first, so the biological system we were always studying was the human red blood cell, which is a very simple cell. And most people don't know that 85% of cells in your body are red blood cells. They're very small, but they're very numerous, critical cells. And importantly, their biology is simple. They, for instance, have thrown out the nucleus, so you don't have to worry about the DNA. So it's a simple biological object or cell to go after. So having said that, I'll say that first in 1989, 1990, very early in my career, we built a whole cell model for the red blood cell based on the available information at the time. So it was first of its kind and showed that you can actually calculate some of the biological properties of the red cell from a computational model, properties you can't see from the individual components. So since then, a lot of time has passed, and maybe the biggest change in this process was the sequencing of the human genome, because once you have the human genome sequenced and you know all the genes, you can take what's called a top-down process instead of bottom-up. It's like you can snap a photo of London from high above and you see the whole system, and you can study it top-down, or you can start looking at a single street or an intersection, which is kind of like a bottom-up. But you need the bottom up and the top down to go together. So the issue with the human genome coming out in the early 2000s, we actually could enumerate the genes that are expressed in the red cell. So we had kind of closure of the biological components you had to consider. So that was easy to do conceptually, 25 years ago. But since then, a lot of time has passed. And during this period, we have now developed very deep data sets about the red cell. So we can now we know exactly which proteins are in it, how many there are. We know metabolism well. And more importantly, we know that the differences between us in terms of genetics affect this cell. So I would say that now, after essentially 4 decades of work, we have what we call a 360-degree digital twin for the red blood cell. And so this is a result of multi-omic integration, as an example, with computational biology. And there it is. It's the simplest cell. We have many other cell types in the body, so there's a lot of interesting things to do going forward. But the lessons with the red blood cell is going to guide us through that process.
Was there a moment in your career where you realized that biology is just too complex for humans to figure out alone?
Yes, I did. And the first kind of, I guess the Germans would call it aha-Erlebnis, happened when I was an undergraduate, realizing how complicated it was. But a lot of unknowns from the beginning. But as I mentioned, once we had the human genome sequenced, we could define it better. And even if you had computational models then, you were missing processes in the red cell. But I would say now that we have all these complex multi-omic data sets that I mentioned, we're getting, not a complete model yet, but it's very comprehensive. So after 40 years of work, we have maybe demystified many functions of the red cell.
Now on to reading the body. A lot of people have done a DNA test, but knowing your genetics and knowing what is actually happening inside your body are two completely different things. Can you explain this difference?
Are you interested in the effects of our individual genetics on our biological performance and how we function? That's what you want, yeah. So we share essentially more or less all of our genes. That's not quite exactly true. Some of us have more or fewer genes from gene duplication, so we may have many copies of the same gene, as an example. And they all have sequence variations in them, which means that they are slightly different in their properties. And so when you, for instance, take your personalized data and mine, you can build a digital twin of your cells versus mine. And we've actually done that about 10 years ago. We had 24 students in my lab, which included me, show up on a Sunday morning in a fasting state, have the blood drawn, and we could do a full metabolomic profile of the plasma as well as inside the cell. And then we genotyped us. And so we actually built 24 personalized red blood cell models. And I was actually quite surprised to see how different people are in some respect. So this was one of the maybe early motivations for doing personalized medicine, because we are all different, and collecting data longitudinally, as a function of age, to see how you actually function and how you might be different than others.
If your genome tells you what you were born with and your biomarkers tell you what is happening today, what do you see when you put those two together that you would completely miss otherwise?
So let's talk about some biomarkers that we are familiar with, like, say, blood sugar level. We have all had them measured, and we know when it's good or bad. We take blood pressure and so forth, the so-called vital signs, about 5 of them. And physicians use that to judge the state of your body and your health. But, we could measure thousands of other variables. For instance, we can measure maybe hundreds and hundreds of metabolites in your body, but they are individual measurements, and they may be slightly different from person to person. But for that to be put together to help you understand what your physiological state is it normal? Is it pathophysiological? You have to integrate all this data together. And you can do that through data models, like these multi-omic data integrations. You can do it through a computational model. You can do it through both, putting them all together. So hopefully that difference between looking at individual measurements and the way your body actually functions will now, I wouldn't say solved, but be resolved or be helped by all these technological drivers we now have available to us.
What does multi-omics mean, and why is everyone in science talking about it right now?
Okay, so what I've been saying so far is maybe jargon, maybe specific to biology, but we can use analogies to understand where we are and where we can go. And I mentioned taking a picture of London from the top and seeing the traffic, seeing the street maps, or I can look at individual streets and potholes or what have you in them. So what I would say here is that, well, first of all, we understand that the street map of London is a network, right? How everything is tied together. But the traffic on that street map is different at 9 a.m. versus, say, 9 p.m. or at midnight. So we distinguish between structure of biological networks like a street map and its activity state. So to push that analogy a little bit forward, your liver has a set of enzymes that metabolizes the food. And what these enzymes are doing is very different, whether you're in a fed or fasting state, or whether you're fed with carbohydrates or proteins or fats. So the functional state is different when the structure is essentially the same, unless you have some genetic lesions. So multi-omics, so let's go back to the traffic. You can be measuring different parameters or different attributes of the traffic when it's happening. You can count the number of cars every minute they're coming down the street, but that may be affected by the condition of the street. Does it have potholes or not? Or the weather. Is it going to be rainy and slippery or snowy and so forth? So there are many, many different data types you can collect that we understand and deal with in terms of traffic. The same thing is going to be true with the biological networks. Now that we have multi-omics, we can measure concentrations of your metabolites, your protein, and you can just measure a lot of things at the same time and comprehensively, meaning a lot of variables. And the challenge that the community is dealing with now is to try to integrate them together into a coherent whole. And this is not easy to do, as you might imagine. So probably one of the defining challenges of 21st century medicine is to make this happen.
And is it true that disease can start in your body years before you feel any symptoms?
Absolutely.
And if so, can we actually detect that now?
Okay, so let's take examples. We all know melatonin as an example, right? So when you're 18 to 20 to 25, you have a very high level of melatonin, but it drops over time, and that's a natural aging trajectory that happens. So if you have been collecting data on yourself over time, called longitudinal data sets, you can see the trajectory you are on, and there is a kind of a reference, healthy trajectory that we are on. And if you're on it, you're aging in a healthy way, and there is maybe no underlying disease developing. But let's go back to melatonin. If you're monitoring somebody that in their, say, 20s, early 20s, all of a sudden sees the melatonin concentration drop way off that trajectory that we would call healthy aging trajectory, something has happened. But you may not realize it because it's very subtle. It's hidden in all the data. And maybe 15 years later, you will develop severe insomnia or something like that, which could have been prevented if you had known about that early subtle change. And that's kind of this notion of longevity research, is to monitor multiple data types at the same time and make sure you're on a healthy trajectory. And if you're slipping off of it, something has gone wrong, and you may detect, cancer, diabetes, and things like that, while you can still treat it early on with relatively mild methods, and you don't have to wait for the full manifestation of a disease that may put you into the hospital or require surgery or something like that. It's kind of a major intervention. So there is something happening in your body. It's complicated at all times. And most of it you're unaware of, like this drop in melatonin concentration. You may not know about it, but it manifests later. You can almost draw an analogy, say, with if you have a car, if you take it for maintenance every year, you can see, how to maintain your car to prevent things. And let's say we take, your tires, they get thinner and thinner. And if you don't replace them, you may have a serious accident on the highway one day. You may slip off the road in the snow or under wet conditions, or the tire may just explode on you. And that's the kind of the way we run medicine today. You wait for a disaster to happen, and then you get treated. But if you're doing multi-omic profiling of you over time, it's like doing a regular maintenance on a car to prevent that accident. So you change your tires, preemptively before you have that kind of accident. So that's how we think about personalized medicine, longitudinal data sets to monitor your healthy aging and detect if something is going wrong, so we can actually intervene early and cheaply, and not end up in the car, in the garage, and you in the hospital or something like that. Maybe that's a helpful analogy, maybe not.
Now on to AI and preventative medicine. So you've been working with biological data and computation for decades. Now that AI has exploded, what has actually changed in your ability to understand human health?
The use of AI and complex data models in medicine and biology is just beginning. But we can say a few things about what we're learning. I don't know if you ever tried to use AI. When you start to, if you're writing, like say in my case, a scientific article, it takes days and days, and you're writing introduction, you're doing this and that, and in your head, you may be thinking about the 3, 4, 5 key papers in this field, and you're describing that. And most human brains can track 3 or 4 or 5 variables. So we are good at comparing A and B, but then C comes in, and that's a little confounding, but we can maybe deal with that. Then all of a sudden there's a D, maybe an E, and it gets more and more complicated to keep it all in your mind. So the best people at keeping multiple variables in their head at the same time are grandmasters in chess. Apparently they can monitor 7, 8 things, multiple steps into the future. So they sit there and think, but they can do it. So we do that normally when we're dealing with writing something or doing experiments. But with AI, it has this remarkable ability to, at the same time, think about thousands of things simultaneously and put them all in context of one another. We can't do that. And I tried to write a couple of articles and asking it questions, and you're astonished. It's just aware of all of these things at the same time. They can call it out in a matter of seconds and change it and modify it and so forth. So the scale at which you can develop understanding or monitor things that are changing is completely different. Like I said, we have 5 variables or so we consider to be vital signs, which are just very basics, but now we can maybe get thousands of them. So how is a physician or biomedical researcher supposed to keep that in their head at the same time? You can't. But AI promises to make that happen. And I think you may have some more questions about the nature of how AI works and how we deploy it. But it enables us now to operate on an unprecedented scale of considering simultaneous things happening in the body at the same time. And an example of that is this multi-omic data integration challenge you mentioned earlier.
Can you give us a real example of something AI can spot in your biology that even the best doctors would miss?
So that's a little loaded question, because to really answer a question like that, you need to have a clinical trial and maybe have a diagnosis or treatment recommendations. So I don't think we are there in any particular case, but we are headed in that direction. So I can give you just one example from the work that we are doing. As we all know, especially after COVID, infectious disease is a serious problem. We have antimicrobial resistance developing that people haven't talked much about, although with COVID, people are more aware of this now. We are now, so do you remember during COVID when you saw these lineage trees, new strain came from South Africa and one from Japan or something, and we were monitoring how it adapted and changed? So we can now do this for all human bacterial pathogens. And E. coli, that I think everybody knows about, can have pathogenic versions or benign versions. So now we are sequencing thousands, tens of thousands, maybe soon hundreds of thousands of these genomes, and we can compare them. And it turns out that unlike us, which share most of our genes, bacteria have a lot of horizontal gene transfer. They exchange genes. Each strain is slightly different. They have a lot in common, but then they're slightly different. So we, for instance, been looking at pandemic strains of E. coli called B2 strains, B as in boy, B2 strains, that's a certain subgroup. And by sequencing them fully, calling all the genes and comparing them to the non-pathogenic strains, we've actually isolated some of their properties. They have unique metabolic properties. They have unique adhesion properties that makes them locate in the infection site, as an example. And we can then analyze those differences. And I can give you one specific example, if I'm not going to use up all the time, like a professor talking and talking. But we analyzed in Denmark 500 urinary tract infections, so UTIs, which are very common. And surprisingly, it's become clear in recent years that the E. coli is responsible for about 70% of these infections. So we took these 500 strains and thousands of other strains and defined what's called the pan genome. So that's the collection of all the genes in all the strains. And not every strain has all these genes, just a subset of them. And we could look at these 500 cases in Denmark. And we had talked about multi-omic data or different types of data. We had one clinical variable to map onto all this genomic data. And that's whether the patient with the UTI became bacteremic or not, which is very dangerous because you can go into sepsis and even septic shock. So a subset of these 500 strains caused sepsis. So we could take those genomes and compare them to these genomes. And sure enough, we found genes that were 10 of them that were unique to those that caused sepsis. And so that was kind of the AI to sort this out. But then we could see what these 10 genes actually did, or 9 out of 10. One of them we didn't have a function for. And 6 of them form a pili, like a hair on the outside of the bacteria. And that allowed it to attach to the bladder wall. And then it had other genes that were known virulence factors. And 2 of them were enzymes that allowed the bacteria to digest its way through the extracellular matrix in tissue, so to get across the bladder wall and into circulation. And then it had another virulence factor, a gene that was a protein that binds to the neutrophils, part of the immune system. And it binds to the so-called apoptosis receptor, which is the suicide receptor that tells the cell to die. So this pathogen would kill off the neutrophils that are coming their way to eradicate them. So this is an example of where we could get very detailed mechanistic information about how a disease progresses from big data sets. And if we had more time, I could give you more of these examples, but they're growing and they're beginning to prove that you can do what you asked. How is AI helping? So in this case, it would give you directly these 10 genes as markers. So if you get a UTI, you would look for them using some detection method like PCR, polymer chain reaction. And if these genes are in you, in your strain, that's a red alert. They would be treated very seriously. And if they're not there, well, maybe you would be more casual about it. So that's one example where big data integration, mostly genetic data in this case, and minuscule clinical data, could give us that answer, identify these, I would say, let's call it threatening genes or dangerous genes that were in your particular strain, in your bladder. And your physician, if he or she would know, they would treat you very seriously right away. One example of many.
Great example.
These will be coming out in the future. And this is something we call actionable information. You can actually act on this one. AI models often just give you statistical parameters and you don't know how to act on that information. But in this case, because we had the knowledge underlying the difference, we could explain it and we have a mechanism to work with. So it's actionable information.
The whole idea of preventative medicine is that we catch things before they become serious. Is that actually possible right now, or is that still a future promise?
Yeah, okay, so this is a very good question, very pertinent right now because of the promise that personalized medicine holds. I don't think there are many concrete examples now in the sense they've gone through a regulatory review and are actionable in the clinic. But there are many studies that are very strongly suggestive that this is possible. So there have been a few wellness or anti-aging type companies that have come out. Most of them have failed because of the expense of generating the data, but the cost is coming down now. So maybe we'll see more examples of the type I'm going to mention now. But some of these companies lived for maybe 5 years with 5,000 patients or so they could monitor over 5 years, they could monitor changes in the physiology of the participants. And there were just numerous examples of that came out of that data set. Like one of the participants loved to eat tuna belly at his favorite sushi restaurant twice a week. His mercury level was way out of line. So they forced him to stop eating tuna belly, and his mercury level came down. So that's kind of one simple, actionable example. And there are many more of them that detect, in particular, kind of kinks in this aging curve of different metabolites in your blood. They should go like this, and all of a sudden somebody went like this. And you could trace the source and maybe treat that just with nutrition. Not clinically treated, but try just to intervene by not eating tuna bellies, type of recommendation. So this is happening, and there are many promising initial examples as those that I gave you.
And a lot of people would hear this and think, I don't want an algorithm making a decision about my health. What would you say about this?
Oh, yeah. I wouldn't want that, would you? Well, so let's talk a little bit about that. So the most common algorithms in AI are these large language models. Chat, Claude, Grok, etc., a bunch of names. So what do? They take all of Shakespeare, all of the articles in Times, New York Times, they go into the Library of Congress, and they build this incredible data set. And then they run their algorithm, whose goal is to predict the next word in a sentence. And the more data they have, the better they can make that prediction. But the bigger is the model is, and it's a statistical model that may have now up to 1 billion different parameters. So you can't really tell what's an association and what is causation. So in medicine, we need to know causation. Okay, so if you take an example in drug development, you have to have a mechanism of action to get a drug approved. And for instance, we can pick an example, say the statins. Okay, people have too high cholesterol level. So people have identified one of the early enzymes in the synthesis of cholesterol. And now I'm going to have to use some jargon. It's called HMG-CoA reductase. Okay, but that's the so-called target. So you want to find a drug that diminishes the activity of these enzymes, and that's what statins do. So if the activity goes down, the whole biosynthesis process goes down, and your cholesterol level goes down. So that's called mechanism of action. So you really need to get mechanism of action. Most AI has no explanation. It just has this correlation. So now people are trying to develop what's called explanatory AI, so XAI. And in biology, you almost have to have it to understand the biology, to justify, drug discovery, biomarker discovery, all sorts of things, unhealthy aging. So you have to have mechanisms. So a number of us are trying to do something that is called knowledge enrichment, where you map knowledge onto the data, and then when you find the correlations, you can use the knowledge to get explanation. And the example I just gave you with the urinary tract infections is an example of that we found these 10 genes, and we knew what most of them did, so we could explain. And then what was going on, how the virulence developed, and how the infection became bacteremic. So now we know what these genes do. So now we could maybe develop a drug that binds to that villi and prevents it from binding to the bladder wall, as an example. So that would be a mechanism of an action and an intervention that is rationalized based on that understanding. So the more examples of that we have in AI applications to medicine, the more we're going to be comfortable with using it. But AI and explanatory AI are two different things. So just to finish that off, we've all heard that AI can hallucinate. It can make bad predictions. They're just wrong, provably wrong, because the algorithm can't distinguish between what's right and wrong. But soon if you have explanatory AI, you put boundaries on the answer based on what you understand the process to be like. So that's what knowledge enrichment actually does. It confines the answers that the algorithm gives to biological reality. And the more knowledge you can put in there, the better that becomes.
Okay, great. Now, what's to come next? So you have worked with some of the best institutions in the world. Most recently, you've worked with Phenome Longevity. So what drew you to that?
What's my read of that? Well, Phenome is a pioneering company. So before I mentioned that people had tried to do this before, but they failed because of the costs. So one of the remarkable things that Phenome has done over the last 4 or 5 years is to be able to put together laboratories around the world that generate data at an acceptable cost. And then now on top of that, they have all these multi-omic data algorithms, and they use some of the systems biology computational models we talked about, which is going to interpret all this data and can give you personal advice based on affordable data, which is the key, and then very rational knowledge enrichment-based assessment of what you can learn from your data. And they also, Phenome has this remarkably good software platform, both the iPhone app and the portal on the web that allows the users to access their own data and look at it. Maybe most people don't want to do that, but some people may want to do that and understand what the data tells them. And remember, I talked earlier about actionable data. So many of the things that happened in the past in this field gave you mutational data or something and some interesting information with it, but it wasn't actionable. You didn't know what to do with that data. It was curiosity. But company like Phenome, and Phenome looks like it's going to be the leader in this field, is actually giving you a lot of actionable information as a result of what they provide. And now at an affordable price, which means it's going to get adopted.
And if all of this keeps going the way it is, what does a normal person's relationship with their own health look like in the next 10 years?
Well, it becomes your responsibility, right? You will be monitored. You will be told how you're deviating from a healthy trajectory. And let's pick something, I don't know, you drink too much and this is all going out of alignment. You're beginning to develop a fatty acid liver disease or something like that. And so you will become probably more and more a participant in managing your health and making decisions on how you're going to react to all this data. And that's going to be, you go to the doctor always decides, you go home and you get your blood sugar level or something. But now you're going to get actual information about your physiology and how it may be changing and what the responsible thing to do is. So it's going to have it's going to take a while, but it's going to have a broad impact on the healthcare system, but also on you as an individual, your lifestyle and how you behave and how you take care of yourself, which we all want to do as well as possible, I think.
I'd say so. And to close, if someone watching this has never thought about their health beyond the occasional checkup, what is the one thing you would want them to know?
You should think about that car that went off the road and crashed. You want to do prevention. You don't want that to happen to you. So as this becomes available and it's affordable and so forth, I think a growing number of people will participate in this. As soon as they are comfortable with that, they get actionable information back at a reasonable price. So it becomes almost like a consumer type issue. No, I think we did good.
I think so.
Thanks for guiding me through this.
That was excellent. Good, yeah.
Are you saying that on the record?
I think I'm on record right now.
Yeah.
Okay, so we'll be pressed.
Good.